Hello everyone, I'm Arzu, and today I'm going to talk about photoimmunotherapy and its anti-cancer effect. Here is an overview of my presentation. First, I will introduce photothermal therapy and different type of photothermal agent. Then I will explain a molecular mechanism of PTT in depth, and then we will see what is immunotherapy and its available immunotherapeutic methods. And then I will show you how combination of these two strategies together has a significant effect in cancer treatment, more specific in metastasis inhibition. Let's start with photothermal therapy, which actually is an encouraging therapeutic strategy that usually used to treat localized cancer. This approach is based on the absorption of near-infrared laser by a photoabsorber that can convert absorbed light to heat and locally destroy tumor cells. But it keeps the temperature of surrounding tissue at a normal level. The study showed that if the generated heat increased temperature of tumor microenvironment above physiological temperature, it produced this destructive effect on tumor cells by several ways, like protein denaturation, impaired DNA function, rupture of cellular membrane, and all of these cause cellular death. So as you can see, compared with the conventional therapeutic modality that we know, PTT could be more targeted to tumor cells rather than the healthy tissue. That actually can be very helpful in reducing negative side effects of cancer therapies. That is the main concern. So as I mentioned, this method involves administration of photosensitive agents, but what they are and how they work. Ideally, these agents should be able to fulfill 4 critical requirements. First, absorb strongly in the near region, but they should show low fluorescence quantum yield and low singlet oxygen generation, because in this way they can maximize the conversion of absorbed photon to heat. Also, they should be non-toxic in absence of photoradiation, because in this way they are selective to the cancer cell just under condition of near IR radiation. Also, they should be biodegradable. It means they should degrade quickly after use, so it provides safety. And at the end, they should be easy to prepare and modify. There are several examples of this material, like metal nanoparticle, mostly gold and silver, organic compound like indocyanine green, and carbon-based material like carbon nanotube, carbon nanodot, and graphene. And also this material can have a different structure, like solid or hollow, or different shape, like nanorod, spherical. Also, they can be coated with various surface coating, like polymeric coating, peptide, hydrophilic, hydrophobic pattern. That's all dependent to their application. But the question is that how this photothermal agent can generate the heat. On the right side here, you can see the Jablonski diagram that show when these photothermal agent absorb light, electron change from ground state to excited state. If the electronic excitation energy relaxes through a non-radiative decay channel, heat is generated around this light-absorbing material, and the result is cell and tissue damage. So now that we know what are these materials and how they work, now the question comes, how much heat should be generated for this method? That actually is a very important question, because we need to consider that treatment effect is very dependent to treatment to temperature range. Here you can see the Goldilocks zone of PTT, which show the temperature threshold for biological responses. Generally, this kind of therapy include 2 techniques, hyperthermia and thermal ablation. That the difference between these 2 methods is the threshold of temperature. In hyperthermia, the temperature is based on this Goldilocks figure is in the fever and heat shock range. But in the thermal ablation, the temperature is more than 43 degrees Celsius, and it's usually mainly is in the cytotoxic range. So as you can guess, the cellular mechanism of each of these should be different, and they actually has a significant effect in treatment. So I will discuss more about that later. But what we need to know that we need to ensure the treatment temperature remain in a desired range, which is based on the literature, something between 40 to 60 degrees Celsius. So you might ask why this range. I will explain the reason for that later. But now just having your mind that it is necessary to carefully control the amount of generated heat to reach this range. And how we can several factors like time of laser radiation, concentration of photothermal conversion agent that we are using, and laser power. So now we know by changing these parameters, we can control the produced heat and increase the temperature of tumor microenvironment to the therapeutic range that can destroy the cells. So I thought maybe it's interesting to know in this range of temperature what happened for cell. So let's see what is the cellular death process. As I mentioned, it's important to understand actually the kinetic and mechanism of this cell death by this technique, because mechanism of cell death activate different cell signaling pathway that has an effect on treatment success. The possible way of cell death that's known by PTT are mainly necrosis and apoptosis. Necrosis here, you can see, is morphologically characterized by evacuation of the cytoplasm, swelling of organelle, and also plasma membrane rupture. That all result cellular collapse. When it happened, like the cellular collapse happened, and membrane, loss of membrane plasma happen, and intracellular contents like damage-associated molecular pattern, that's called DAMPs, release into extracellular environment that can cause immune system activation. But what about apoptosis? So the cell membrane integrity is maintained during apoptosis process, and it may signal like phosphatidylserine relocate to the extracellular portion of the membrane to mark the cell for phagocytosis. Also, this event is associated with the release of anti-inflammatory signal, like transforming growth factor beta, that actually this has a suppressing effect and inhibits cytotoxic T cell activation. So at the end, it avoids immune activation. So we can say apoptosis is considered as an immunologically silent type of cell death. But if the phagocytes do not rapidly clear an apoptotic cell, it can also experience loss of membrane integrity and release its intracellular content, including DAMPs. And this process is known as a secondary necrosis. That is here. Now, you might ask how this pathway happen and how we can choose each of these since they have a different effect. So generally, I can say high energy irradiation can cause a necrosis, but low energy radiation, I mean low temperature, can promote apoptosis. And as each of these ways has a different biological response, I thought it helpful to see how experimentally we can determine each of these after laser radiation. In the right side, you're seeing the figure of the cells that has the green and red fluorescence. The green part is a live cell, and the dead cell showing the red fluorescence. And it's just a spot that laser radiated. Also, this figure is clearly showing that how selective is this method in killing the cells. But how we can find this live and dead cell? Typically, the cell death characterized by two different types of fluorescence dye, that is calcine AM and ethidium homodimer. The mechanism is when the cells are live, the non-fluorescent calcine AM molecule is converted to green fluorescence after it's hydrolyzed by intracellular esterase. Because dead cell doesn't have this active esterase, so just live cell can show green fluorescence. But how indicate the dead cell? We can use the ethidium homodimer that is impermeable to cell with an intact plasma membrane. But when it loses its membrane integrity, it can permeate to the cell and bind to the DNA, and after bonding, it emits a strong red fluorescence. So in this case, we can see that the difference of red and green is a live and dead cell. So, but although these molecules can show cell death after PTT, they cannot determine loss of membrane integrity that I mentioned is because of necrosis or apoptosis or even secondary necrosis. So for this purpose, there is another way that is annexin V and 7-AAD that's used to determine apoptotic and necrotic cell. Annex V is a protein that is specifically bind to phosphatidylserine that I mentioned before on apoptotic cell, and 7-AAD is a fluorophore that interact with DNA and used to analyze loss of membrane integrity. So if we have these two a marker, and it shows Annex V positive, for example, and 7-AAD negative, it's showing that most of cells are underwent apoptotic. And if it's both positive, cell are secondary necrotic cell. So also I brought a TM image of each of these process. You can also image them and see what's happening for them. So now we know how to characterize cell death pathway. I'm going to show you what other information about PTT effect we can obtain by this type of staining. Here is one example. I don't want to go to detail of this paper, just I brought one part of that was very interesting, that's showing that how the temperature has an effect on PTT efficiency. Here they treated cell with gold nanoparticle for a certain time, and then they irradiated it with a laser for 10 minutes and monitored the temperature. But one interesting that they did was one time the initial temperature was from room temperature, another time was from body temperature. And after 10-minute laser radiation, data showed 12 degrees Celsius increase in case of room temperature from 29 to 41 degrees Celsius and 13 degrees Celsius from 37 to 50 degrees Celsius for another batch. And in both cases, temperature reached a constant amount. So as we can see, although the rate of temperature are similar, the final temperature are different, about 10 degrees Celsius. So then they find the effect of initial temperature in cell death by analyzing it 5 hours after radiation by monitoring NX3 staining and 7AAD. So result here is showing that clearly cells that were irradiated at room temperature were unable to reach sufficient temperature to cause cell death. At room temperature, less than 10% of cells are dead, but cell irradiated at biological temperature 37 produce around 58% cell death. So we need to consider that the ability to induce effective cell death by PTT is very dependent to a starting temperature of the cell. Also, they did another important and interesting experiment. They check different type of cell death. For this purpose, they have performed a kinetic study after laser radiation for different time, 30 seconds, 2 minutes, 4 minutes, and 10 minutes. And the percentage of both apoptotic, I mentioned NX3 positive, 7AD negative, and secondary necrotic, both are positive, clearly increased at longer incubation time, like 5 hours, 12, and 18. And it shows that cell become first apoptotic, and only later the membrane is disrupted and it's become secondary necrosis. So this was one example, but generally a limited number of studies have explored in detail if cell death was apoptotic or necrosis, and after laser radiation, what happened, and how light exposure condition have an effect on that. So, but I tried to find some example from different studies that included checking effect of temperature in different cellular death pathway. Here is several example. I just want to choose that part of their study that talking about effect of temperature on cellular pathway. Here is one example that's showing that they used the gold nanorod and they irradiated the tumor for 2 different times, 2 minutes and 5 minutes. After 2 minutes of laser radiation, most of cells underwent apoptosis. But after 5 minutes radiation, most of population observed were necrotic cells. And so this result also confirmed what I mentioned before, that a low dose of heat induced apoptosis, but high dose of heat induced necrosis. Based on the temperature, you can say after 5 minutes, temperature was 52 degrees Celsius, and 2 minutes reached to 42 degrees Celsius. Another example is here, that here there was not any laser radiation or photothermal conversion agent. They just checked effect of heat on the cells. And they find that in the temperature greater than 42 degrees Celsius, cells are getting dead. And following heat shot at 44 degrees Celsius, you can see most of cells underwent apoptotic cell death, but in the 46 degrees Celsius, the cells were necrotic. So that was another example. Here you can see another work that they explored the effect of temperature on the cellular death pathway. So for this purpose, they used gold nanorod for melanoma cancer. They reported in a lower temperature at 43 degrees Celsius, it showed the cell killing is not efficient. When the temperature increased to 49, most of the cells are necrosis, and but in the moderate temperature, 46 degrees Celsius, it's mainly our secondary necrosis. So yeah, there were some, this data actually suggests that under different experimental conditions, cell death pathway can be changed. And so the mechanism of cell death is very dependent in the intensity of radiation, the manner of heating, I mean, the slow or fast, and duration of the heat shock. Because all of these that I mentioned can influence the temperature of solution after radiation. So now we know how we can reach to the necrosis and apoptosis of cellular pathway. So let's look a little deeper to this process and see which one is more favorable for cancer treatment. But before starting that, if you remember, I had mentioned necrosis can activate immune system, but apoptosis is not able to activate it significantly. But let's look why. Generally, the cells that die by necrosis release intracellular organelles, antigen, and damage-associated molecular pattern DAMP, such as DNA and heat shock protein. Dendritic cells that phagocyte these DAMPs activate the nuclear factor kappa beta pathway, which then promote the expression of co-stimulatory molecules CD80 and CD86. And then dendritic cells that present antigen and major histocompatibility complex, MHC molecule, also show co-stimulator and stimulate T cell and promote the immune response. But on the other hand, cells that die by apoptosis also release antigen that can be picked up by dendritic cell, but they do not typically release DAMP. Without phagocyting DAMP, the NF-kappa beta pathway is not activated, and expression of CD80 and CD86 is not induced. And without this co-stimulator, T cell energy may occur, so it's actually suppress the immune response. And what is energy T cell? Actually, it happens when naive T cell responds to self-antigen peptide, and then when they want to get activated through binding by B7, which is CD80, CD86, it doesn't happen because there is not any pathogen there, so it remains inactive actually in the body. So by this explanation, I wanted to show PTT can induce both immunostimulatory effect and immunosuppressive effect. And this stimulation or suppression is dependent to amount of necrosis and apoptosis, and the proportion of these actually response. And as I mentioned before, it might change over time. Based on what I mentioned, we can say the necrotic cell death seems critical for inducing an immune response with PTT. See, here, I'm just trying to show that after laser radiation, there is several pathways, but this is the optimal thermal dose that we want that cause the activation of immune system. This here, I'm going to just review it quickly. The result of necrosis is, as I mentioned, is release of intracellular antigen. These antigen are attracted by antigen-presenting cell, like dendritic cell, and then dendritic cell localize into the lymph node where they present antigen to T cell through their MHC complex molecule, MHC molecule, and then activate tumor-specific CD4 and CD8 T cell. And these tumor-specific T cell now then migrate to the tumor site and can attack it, the tumor. So, but as you see, the immune system during this process is getting activated. But the problem is that although this inflammatory activation is happening in the microenvironment, but this is not sufficient to induce a strong immune response. Because of that, so there is a need to enhance this signal. But how? Actually, immunotherapy can combine with PTT to enhance immune response against cancer on cellular level. But before talking about the combinational system, I'm gonna talk about immunotherapy and its different class. Okay, so immunotherapy simply is using our own immune system to fight against cancer. However, cancer cell can evade destruction by this immune system through several mechanisms. is very smart. One way is by secreting cytokines such as interleukin-10 that suppress dendritic cell maturation. We know that mature dendritic cell capture tumor antigen and present it to the T cell for activation. Also, a cancer cell can produce factors such as TRAIL and FAST that induce apoptosis of cytotoxic T cells. Cytotoxic T cells, they are responsible to find and destroy cancer cells, but now they cannot do their job. And another thing is that cancer cells attract immune suppressive cells to the tumor microenvironment, including tumor-associated macrophage, myeloid-derived suppressor cell, and T-regulatory cell. So I have in mind this part because I'm going to discuss about that later more. But based on this, we can see our own immune system is not successful in fighting against cancer. But immunotherapy has been emerged actually to recognize and kill cancer cells that have adopted to these avoiding immune recognition mechanism. But how it does work? Actually, immunotherapy contracts this mechanism by several ways, like delivering tumor-specific T cell, immune checkpoint inhibitor, like antibody against CTLA4, PD-L1, or CPG. That all of these examples are part of different class of immunotherapy, like immunomodulator, cell-based immunotherapies, and oncolytic viruses. But before explaining each of these methods, I'm going to look at the history of this field. Pioneer work in immunotherapy was started more than 100 years ago in 1891 by William B. Coley. He injected bacteria into patient with that has a cancer. He thought that the infection he produced would have a side effect of shrinking the malignant tumor. He was successful, and this was one of the first examples of immunotherapy. Coley injected more than 1,000 cancer patients with bacteria or bacteria product, and he reported excellent results, especially in bone and soft tissue cancer. Because of that, he called it the father of immunotherapy. So now we have some idea about the background of this field. I'm gonna explain each of the available therapy, immunotherapy method. One method is adoptive cell therapy, that's known as a cellular immunotherapy. It's a form of treatment actually that uses the cells of our own immune system to remove the cancer. So some of these approaches involve directly isolating our own immune cell and simply expanding their number, but others involve genetically engineer our immune cells and increase their anti-cancer capability. And there are different types of that, and the one is tumor infiltrating lymphocyte therapy. So I briefly explain each of these. This approach actually harvests naturally occurring T cells that have already infiltrated the patient tumor, and then they activate and expand them. And after that, the large number of these activated T cells are reinfused into the patient, and then they can destroy the tumor. But unfortunately, not all patients have T cells that have already recognized their tumors. In some patients, these T cells are not able to be activated and expanded to sufficient number. For this case, there are another approach known as engineered T cell receptor therapy, TCR therapy. And in this approach, again, they take the T cell from patient, but instead of just activating and expanding the available anti-tumor T cell, the T cell can also be equipped with new T cell receptor that enable them to target a specific cancer antigen. These two methods that I mentioned actually can only target and remove cancer cells that present their antigen in a certain context. I mean, when the antigen are bonded by the major histocompatibility complex. But except that, they cannot work. To overcome this limitation, chimeric antigen receptor has been emerged that in this way, T cells actually are taken from patient blood, and the gene for a special receptor that bind to a certain protein on the patient cancer cell is added in the laboratory. As I mentioned, key advantage of this CARs therapy is their ability to bind cancer cell even if their antigen are not presented on the surface of the cell, which can cause more cancer cell be destroyed during this process. So this was one example of like adoptive cell therapy. But The second class is immunomodulator, that's including immune checkpoint inhibitor and immunoadjuvant. Last week, Alicia explained an immunoadjuvant, so I'm just gonna skip that and talk about immunocheckpoint inhibitor. To explain it better, I brought this figure here that's showing the process of how T cell is activated, then how it gets inactivated, and at the end, how by using this checkpoint inhibitor, we can make it activate again. So to understand that, I'm going to briefly explain each of these steps. So T cell activation, as I mentioned before, is initiated when antigen is presented to T cell receptor, TCR, by MHC class 1 or 2 on an antigen-presenting cell. Signal from TCR then amplified by co-stimulatory molecule and binding of CD28 on T cell to this co-stimulatory molecule, that is B7-1 CD80 and B7-2 CD86 molecule, and the antigen-presenting cell creates a signal that causes T cell activation. This CD28 B7 binding actually is very necessary and causes increased production of interleukin-2 and other stimulatory cytokine and cause T cell proliferation and differentiation. And this is what we want. Also, there are several regulatory checkpoints that normally prevent the inappropriate activation of cell-mediated immune response. But they also are expressed on T regulatory cells that are suppressive cells. One of these are cytotoxic T lymphocyte-associated antigen 4, CTLA4, which is one of these checkpoints. But the problem is that when B7 binds with CTLA4 instead of CD28, it does not produce its usual stimulatory signal. So CTLA4 actually, by this binding, inhibit T cell stimulation and promote the T cell energy that I explained before. So if we can inhibit this checkpoint, the immune system will be free to respond. So this is what these immune checkpoint inhibitors are doing in immunotherapy. Here you can see an antibody that blocks CTLA4 function can inhibit CTLA4 and cause T cell activation. And yeah, this is actually a basic of immune checkpoint inhibitor, but I will show you one of these examples of how they are useful for cancer treatment later. Yeah.
I had a question, if you can go back. So in this image here, I see that you put, these are antigen-presenting cells, but I thought in most cases the CTLA4 was produced on tumor cells, and that's what inhibits it.
Yeah, that's true. I said it's overexpressed in T regulatory cell and yeah, and tumor cell as well. So it's just example, but the mechanism is exactly same.
So the dendritic cells can, or APCs can also express or inhibit that as well?
APC, no, not APC can inhibit that. Like, you know, if there are in the tumor cell, for example, if we had the anti-CTLA4, so their reaction actually is gonna be quenched. Like, you know, it's not gonna work after that. It's not, they cannot be recognized. Did I answer your question?
Yeah, I think so. I guess I just got a little confused because here you're showing an APC, and the APCs looks like it's inhibiting the T cell.
APC is inhibiting T cell. Which part here?
Yeah.
You know, the point is that if this B7 bind to CTLA4, it can, everything that bind to CTLA4, like here is in this APC is B7, it's going to be inhibited. They cannot work. So if we block that part, so now there's not any concern. But we always, you know, that we don't want to block that because sometimes it's useful. But in terms of cancer, because it's overexpressed and it's limit the activation of the dendritic cell immune system, it's good to be blocked.
Yep, thank you. That makes sense.
Okay, so another class of immunotherapeutic method is oncolytic viruses. And normally, we know that we avoid the virus infection because virus can kill healthy cell and cause disease. But some viruses target and infect cancer cell specifically. This virus called oncolytic virus and can be used as an immunotherapy method. So this method actually is on selective replication of viruses in cancer cells, and they're spread inside the tumor without causing damage to normal tissue. That's very interesting, actually. When they enter to the cell through their specific receptor on the cancer cell, continuously replicate and lyse the cell, so the result is killing the malignant cell. Also, this method have many features that make it very advantageous from different therapeutic strategy. One is that there is low probability of generation of resistance, not seen so far. And also they replicate in a tumor selective fashion. Also, they are non-pathogenic. At the end, their dose in tumor increase with time due to in situ virus amplification. All these make it very interesting immunotherapy method. So now let's get back to the PTT after we know all of these immunotherapeutic method. And each of these strategies that I mentioned actually has been evaluated in combination with PTT. And the results show that dual PTT immunotherapy is very advantageous to either therapy alone. And the reason is that we talk about that actually. The PTT is activating immune system through this pathway. And here, so the signal was low. And if we are going to enhance that, if we combine this with like immunotherapy agent, this part is going to be enhanced and activate the immune cell system more and attack strongly to the tumor. So in the next few slides, I'm going to show you how this combination system works and show how this synergy effect enhances the anti-tumor efficacy. Here's the first report and the combination of photothermal therapy with immunotherapy that done by Dr. V. Chen. Their approach actually consists of three components. They use 805 nanometer diode laser, and they use photosynthesizer that was ICG in the cyan green, and also they use glycated chitosan gel as an immunoadjuvant, also as a carrier of ICG. And then he checked anti-tumor effect of different combination of this photo absorber and immunoadjuvant on a rat model implanted with VMBA breast tumor after irradiated it with the laser. They monitor tumor volume and survival rate of each group, and then they reported the optimized combination that is here. They show in this optimized condition 50% survival after 90 days after treatment. Also control was showing that 0% survival in less than 30 days. Then they check metastasis effect of this method by a tumor rechallenge experiment. So to do that, they injected rats with 3 times the standard dose of tumor cell, and they find 50% case there was in 50% case there was not any effect of tumor development after 16 days. That actually was very interesting because it showed a long-lasting systematic effect of treatment. This study appeared actually as an effective approach for destroying a primary tumor and metastasis inhibition, and inspired many other researchers to actually investigate the new combinational system. That I will show you some of those in a few slides. Here is the next example. This is combination of PTT and an anti-CTLA-4 checkpoint inhibitor that I explained the mechanism of that before. So in this work, they use pegylated single-wall carbon nanotube with 200 nanometer length as a photothermal agent that has a strong absorbance in the near-eye region, so it can generate heat after laser radiation. But what they did first was they showed that single-wall carbon nanotube by itself, without any radiation, has an immunological response and can activate dendritic cell and release also cytokine. Here you can see the dendritic cell maturation of single-wall carbon nanotube compared to the control. But also they find that this response is not strong enough to inhibit tumor growth by itself. So after that, they checked how single-wall carbon nanotube-based photothermal ablation of tumor can trigger immunological response in the animal model. To do that, first they injected intratumorally single-wall carbon nanotube into the mice that bearing 41 breast tumor. And after 10-minute radiation, so you can see that the temperature, the tumor temperature increased to 53 degrees Celsius. Now, I guess all of you know that this is a temperature that is high enough to effectively ablate the tumor cell. And then effect of PTT on dendritic cell maturation and cytokine release were studied. Mice were sacrificed at 96 hours after radiation, and the maturation of status of dendritic cell inside tumor draining lymph node was checked by flow CT. So it was found that single-wall carbon nanotube-based PTT treatment, you can see, could increase dendritic cell maturation in lymph node much more than just single-wall carbon nanotube by itself without any radiation. Also, they analyzed various cytokines in serum of PTT-treated mice at 12, 72 hours, and 168 hours post-ablation. And both of these single-wall carbon nanotube and PTT version of that were able to increase the secretion of pro-inflammatory cytokine like interleukin-1 beta, interleukin-12, interleukin-6, and TNF-alpha. After this experiment, they again checked if this immune system activation can prevent the tumor growth, tumor metastasis, but they find that the immunological response induced by PTT is not enough to completely inhibit the growth of secondary tumor. So I will discuss more about that next slide. But then they decided to enhance the anti-tumor immune response with the method that I mentioned before. It's a CTLA-4 blockade therapy. Here is their experimental design. So they inoculated the first tumor by subcutaneous injection of 41 tumor cell on the left flank of each biopsy mice. And 1 week after injection, when the first tumor reached to 150 millimeter cubes, a second tumor was then inoculated on the right flank. And at the following day, the first tumor was intratumorally injected with single-wall carbon nanotube and treated with PTT, or they simply removed the first tumor by surgery. After removal of the primary tumor by either method, mice were IV injected with 3 doses of anti-CTLA-4 antibody on day 1, 3, and 5. And the growth of secondary tumor actually, and also survival of those mice after different treatments were monitored. As you can see at figure B, anti-CTLA-4 without PTT could only slightly delay the growth of secondary tumor when their primary tumor were removed by surgery method. But for mice that their primary tumor were treated by PTT, CTLA-4 blockade could significantly inhibit the development of secondary tumor. And actually, these results show that although single-wall carbon nanotube-based PTT alone could only partially delay the growth of a second tumor challenge, its anti-tumor effect would be greatly increased when combined with CTLA-4 therapy. And this led to effective rejection of secondary tumor, which actually can mimic the tumor metastasis. In figure C, you can see they also checked T cell infiltration in secondary tumor after 10 days after injection. You can see the percentage of CD8 T cell in the secondary tumor after the combined treatment was 3-fold of surgery treatment or PTT treatment alone, and 1.5-fold actually compared to the surgery plus anti-CTLA-4 treatment group. Also, the percentage.
I have a question. Yeah. Specifically about the CD4 CD8 results. What is the significance of actually showing improvement in the CD8 but no improvement in CD4?
Yeah, CD4 actually improved a lot, but you can see PTT here showing the more show more signal. But the reason was that most of this CD4 was T regulatory T cell. I wanted to explain here because they were showing that PTT is increasing the amount of CD4, but most of those helper T cell was T regulatory cell, so it wasn't effective. But compared to that, you can see PTT with the anti-CTLA4 has an effective, actually, CD4. They explain because, you know, PTT, like, yeah, there are a lot, but you can see some part of these are inactive. This is showing that amount of CD4 that are not working because they are T regulatory cell.
Okay.
Arzu, can I follow up on that question? I guess
Sure.
what I wonder is how did they actually obtain the T cells?
How they obtain T cell? This is from that actually this is a I think that this is from tumor. This is from secondary tumor. There's different way. You can get the lymph node, you can get the tumor. It's very dependent on what you are going to show and the time of that you sacrifice the mice and separate this. But for this case, is in the secondary tumor because the only thing that they wanted to show was the metastasis part. So they care about the secondary tumor environment.
Right. I guess I asked because if they would have taken it from the lymph node, there's no proof unless they incubated for 72 hours with tumor cells. There's no proof that these are specific to the tumor. But if they took it from the tumor, then for sure they'll know that it's you know, tumor infiltrating T cells. And so that's where my question comes.
Yeah, and it's in vivo, it's not in vitro. They didn't incubate this T cell with anything. It's just directly from the tumor. Yeah.
So these are tumor infiltrating.
Yeah, yeah.
Okay. And I guess you know, for regulatory T cells, it's I guess it's good that they have it higher for PTT because usually regulatory T cells will suppress cytotoxic T cells and other immune cells from helping out, right?
Yes. Why it's good?
Yeah. I'm just clarifying why is it good that like for example, having high regulatory T cells, is that a good thing or a bad thing?
Oh, it's a bad thing. It's a bad thing, but the good thing is that CTL4, this therapy can reduce that amount.
Right.
Just imagine if it was PTT, we saw it's not effective on metastasis because of exactly this regulatory T regulatory cell or this immunosuppressive environment that is actually causing, and it kind of promote metastasis instead of inhibiting the metastasis. So
Right.
because of that, PTT by itself doesn't work because of T regulatory T cell and this tumors suppressive microenvironment. And but after anti-CTLA4 therapy, it helped and reduce this effect.
Right. Making it a stronger immune response.
Yeah, exactly.
Thank you.
Okay, so yeah, as we discussed, the percentage of CD4 also increased and the secondary tumor of mice. And I explained this in the figure D. You can see PTT alone. Most of these increased CD4 T cell are regulatory T cells, which are actually able to suppress effective anti-tumor immune response. But as you can see, these results showing that this combination therapy not only increased the amount of CD8 positive T cell, but also it increased effective ratio of CD4 T cell to T regulatory cell. Last thing that they checked was after this was lung metastasis. They did that by challenging mice with IV injection of 41 cells. Here you can see the photo of India ink stain whole lung and H&E stain lung. Compared to mice with only surgery of the primary tumor, those after PTT treatment and anti-CTLA4 therapy are showing reduced lung metastasis. Also, after that, they did survival of mice treated with this combination therapy. You can see that around 57% of mice survive in 50 days after combination therapy, but just only 25% could survive when this therapy done with surgery in 50 days. And none of mice actually survived with surgery or PTT alone. So these results kind of showing that combinational system is able to induce a strong anti-tumor immunological response to inhibit lung metastasis and also increase the animal survival time. So here is another example of combination of photothermal ablation and immunotherapy, but this time is between PTT and immunoadjuvant. So in this work, they develop an endogenous vaccine-based system they use IR7 fluorophore loaded liposome that actually coated with immunoadjuvant that they use CpG. IR7 is a new IR dye with high phototherapeutic efficacy after laser radiation. And CpG is immunoadjuvant that potentiates the immune response to an antigen or to the desired immune response, and they bind to toll-like receptor 9 of antigen-presenting cell and enhance the expression of co-stimulatory molecule and releasing different inflammatory cytokine. So this is their system here. Then they check, they wanted to check the PTT effect after the tumor size reached to 100 millimeter cube, and they injected it with 5% glucose as a control and dye conjugated to the liposome, and also IR7 liposome coated with CpG. And they radiated it for 5 minutes. So as you can see again, after laser radiation, mice treated with IR7 liposome or IR7 liposome with CpG show localized heating in the tumor area. And temperature in the tumor area, you can see increased to 50 degrees Celsius. And they monitored the tumor size in 2 weeks. And you can see here, data show that tumor growth after combinational therapy plus laser treatment was much slower than the mice that treated with each of these therapy, PTT or immunoadjuvant alone. That again, it confirmed that combination of PTT and immunotherapy can provide a significant benefit for cancer treatment. Here is obviously we can see the tumor. Then this part is same as the previous paper. They evaluate, they actually check the immunostimulatory activity of this combinational system. First, they study effect of PTT with the immune system, this combinational actually system on dendritic cell maturation in vitro. So this is the way that they did bone marrow dendritic cell were isolated from mice and then incubated with this combinational system, IR7 liposome with CpG and that for 24 hours. And the expression of co-stimulatory molecule like CD40 and CD86 was significantly increased on bone marrow dendritic cell that treated with this system after laser exposure compared to other treatment. So as we discussed before, that tumor cells that are located in the immunosuppressive microenvironment that contain large number of like T regulatory cell, myeloid T cell. So they did an interesting experiment and they they did a secondary tumor experiment and they checked these cells, suppressive cells actually. They choose T regulatory and myeloid cells. The mice were sacrificed 48 hours after injection and the tumor removed and the immune cell population inside the tumor microenvironment were tested by flow CT. Here is actually the marker for myeloid cells that's showing that the percentage of that is increasing. It was about 3-fold compared to the control group when they had just the live version here. And also they checked the T regulatory cells, that the marker is FOXP3, and they find here that it was 7-fold more for PTT compared to the control. And these results showing that by using the immunoadjuvant, this mechanism is getting reversed, and the immunosuppressive tumor microenvironment that induced by PTT is getting less. So it's a kind of showing that combination of PTT with immunoadjuvant is helpful for cancer treatment. The last example is between photothermal therapy and adoptive immunotherapy. So they show the inflammatory and anti-tumor immune response for the melanoma, yeah, the skin cancer cancer. And in this work, they used the 40 nanometer gold nanoshell as a photothermal agent and combine it with adoptive T cell therapy. So here is exactly the same as what we talked before. They find that PTT can promote the expression of pro-inflammatory cytokine and chemokine. So for this purpose, a serum were isolated from PTT-treated mice 24 hours and 96 hours after radiation. and serum cytokine and chemokine analyzed. As you can see, PTT induced expression of pro-inflammatory cytokine like IL-6, TNF-alpha, interleukin-1 beta, and IL-12. They also increase. Also, PTT can induce cytokine that stimulate stem cell survival, growth, and differentiation. They are GM-CSF, MCSF, GM-CSF. That actually they show significantly increased following the PTT. Another thing that they checked was the amount of chemokine that is increased. CXCL1 actually is responsible to attract neutrophil to the burn site after radiation. It was significantly higher 24 hours after radiation. And CCL2 and CCL4, they attract monocyte, macrophage, dendritic cell, and they also significantly upregulated at the same time point after 24 hours. Also, they determine if PTT activate dendritic cell. For this purpose, the maturation of dendritic cell inside tumor draining lymph node was checked by flow CT. And you can see it's showing that compared to control, PTT enhanced it. Also, the amount of CD8 and CD4, same as before, they check it, and it's showing the increase for the PTT group. Actually, I like the way that they reported this data. And also, again, they check the myeloid T cell, that is a suppressive T cell, not T cell, suppressive cell, and they find that the PTT again is increasing the amount of immunosuppressive cells. So after that, they check also the tumor metastasis is not working just by PTT. You can see here very obviously. So this is a primary tumor that is PTT is able to actually control it. But the secondary tumor, you can see it's growing like as a control. So then they decided to actually combine this PTT with adoptive immunotherapy. So here, first they check that the potential effect of PTT on adoptively transfer PMEL T cell that are specific for melanoma tumor antigen GP100. I should mention PMEL1 T cell came from transgenic PMEL mice that actually express a single transgenic T cell receptor that recognize the GP100 that this antigen overexpressed in melanoma. So this is their actually T cell that they're going to inject. The primary and secondary tumor then established, like as before. Primary tumor, well, actually the primary tumor ablated by PTT on day 10, I guess. And 24 hours following ablation, this PMEL T cell were infused through IV injection. And then they measured the frequency of the T cell in a spleen and tumor draining lymph nodes. 9 days, I think, a week after adoptive transfer. They did that by measuring INF gamma because that is released in response to the GP100 that is overexpressed in melanoma. You can see here the highest number of the tumor reactive PMEL T cells is in the spleen and lymph node. In mice is belong to the group that treated with dual PTT and this T cell therapy. This data also suggests that PTT can promote PMEL T cell expansion. Because if you compare it with just PMEL T cell, you can find it. Also, then they check tumor recurrence, and they find that the second tumor that was established on the opposite flank of mice, you can see here, show that when they treated the tumor with PTT alone and PTT with this method, it's for the PTT alone, it's recurred in around 35%, compared to only just 13% in mice that got these two therapeutic methods together. And then they checked the lung metastasis, and same as before, you can see that it was effective. Number of lung metastases was less for this dual therapy compared to each of therapy alone, and also lung mass was same. So yeah, so I hope that I was able to show that. I actually was able to show that how photothermal therapy work and look at the cellular death and immune response activation of this method. And then I show that what is the immunotherapy and what is a different type of that. And also I show that how the combination of these two strategies together has a significant effect in the metastasis inhibition treatment. That's all. Thank you. If you have any question, I would be happy to answer.
Thank you very much, Arzu. That was very comprehensive and covered a lot of pretty good research that I hadn't even seen before. So, does anybody have any questions?
Hey, Arzu.
Oh,
Go ahead, Tom. You can go first.
okay. So Arzu, can you go back to slide number 10? Yeah, here. So you mentioned that 3 different types of thermal dose. And in case of the too high or too low, you mentioned it as like non-immunogenic cell death. Can you please elaborate this term a little bit? Like, I mean, what do you mean? What do you mean by the non-immunogenic cell death here?
You're muted, Arzu.
Okay. Sorry. Yeah, your question is that what is non-immunological cell death? So if the thermal dose be too high, actually after necrosis that happen, it kind of aggregation between the cell is happening and the mechanism of cell death is not going through the this pathway that I showed, the activation of the T cell or immune system. So it's going in a different way. Like if it's with too low, I mentioned it's going through the apoptotic pathway. That way the immune system is not activated. So here I think I explained that how apoptosis is happening, and yeah, here. So there is different way that they can be die. If it's not necrosis that usually release dams and go through the immunological response. These this pathway, apoptosis, is going through the phagocytosis and other pathway of cell death that is not related to immune system. And also, I mentioned if it's be too high, so usually aggregation and these things is happening, and it's not again releasing the dams, or kind of it's releasing, but it's removing that dams as well because the temperature is too high. It's usually, yeah, it's if the temperature goes high, they're not effective for activation of immune system.
Okay, thank you.
Hey, Arzu. The talk was fantastic. I think it has my mind with a lot. It filled my mind with a lot of wonder, I guess. And what I'm wondering, and I guess what I'm wanting to get is your opinion on what you think is a better form of therapy. Then would you do PTT with like how you're doing with viruses, or do you think that maybe doing immune checkpoints with PTT is a better route?
I don't think a virus by itself. I hope that it showed the like an immunoadjuvant effect, but actually immunoadjuvant was not that much strong, that checkpoint inhibitor and also adaptive T cell. But if virus work as same as immunoadjuvant, I'm saying still it has a problem, but I'm not sure that virus is that much strong too. Compared to CPG. So we should see that part. But based on what I read, I think we need to combine this with another treatment if we are gonna care about the metastasis. If we put our purpose mainly on the activation of immune system and primary tumor treatment, I think we are fine, and it's much better than, it might be much better than PTT. But if you look at the metastasis, I'm not sure that how successful this is gonna be.
I guess like based on all the literature you presented, I feel like one of the main issues is having to break through this immune suppression. So I feel like if you can just, whether the virus or CPG, I think as long as you're able to break through all the suppression that's going on with regulatory T cells and maybe even I think M1 or M2, I think I can't remember which one. I think it's M2, right?
Yeah.
Yeah, I feel like if you can just, I feel like that's what CPG is doing. It's probably just allowing the macrophages to change into the correct phenotype and allowing for the immune system to, you know, break through and allow for cytotoxic T cells to come in. So I think, and I guess that's where my mind is going, is that I feel like it's just breaking through and making it like I think PTT by itself, it looks like it's already able to activate the immune system, but because of all these suppressors, it seems like it reduces the response.
Exactly. Yeah, it's happening. But I was trying to kind of compare this immunotherapy method together to find which one is better. I'm not sure, I couldn't find a reference for what I'm saying now, but I feel that immunoadjuvant is compared to other therapeutic method is less effective based on the data, because they are, still we can see the tumor here. So, and it's like one tumor, it's not the secondary tumor. So it's like, the primary tumor. So it might be not that much.
Right, and that's what I'm saying. I feel like that's just, I think that's just aiming for APCs. It's not really aiming towards regulatory T cells.
Yeah,
And
true.
so with the immunocheckpoint therapy, you're actually allowing T cells to come in, and so I feel that's the reason why it's a stronger response.
Yeah, that's true. I agree.
Yeah, pretty interesting. I think it was a really good talk.
Thank you. Any more questions?
Any more questions? Okay. Well, thanks a lot. At some point, we have to turn this into a grant and also a paper. So let's do all those things. Okay, so everybody, clap in front of your monitor.
Thank you.
All right, very good. Okay, well, I guess I'll see you guys on Wednesday, okay?
Are you sure not Friday?
No, I got rid of all the Friday meetings.
Wait, I thought that was Scribble Day.
We could do Scribble on Friday again. That was fun.
Yeah, that was,
Yeah,
that
you
was
guys
awesome.
missed out.
Yeah, you guys didn't show up for Scribble. It was really fun. Losers.
Yeleni's a great drawer.
No, she's not.
She's way better than I was.
It's true. I'm still stuck on the saxophone she drew.
All right, folks. I'll see you Wednesday.
You guys have a good, take care.
Bye.